All right, another thing that we oftentimes need to do if we talk about techniques is there needs to be a way to multiply the DNA, make it more abundant, and we need to do that in vitro. So again, under artificial conditions inside of a test tube. And the most common way that this is going to be done is with this technique called the polymerase chain reaction. Well, what the polymerase chain reaction is going to do is it's basically going to take the process of DNA replication and it is going to artificially create that, not only just copying and making DNA, but we're going to specifically copy just one little region of the DNA. So here you can see that we're going to start with maybe one copy of the DNA sequence that we want. We're going to run a cycle, we get 2 copies. We run another cycle and we get now copies of each of those. So every round that we do, or every cycle of DNA replication here in our test tube, is going to basically exponentially expand the amount of molecules that we do have, or the number of copies. So with PCR, the benefit of this is that we can start with a single molecule of DNA and we can generate billions of exact copies within just a matter of hours. So there's great potential for this. If we look at the overall process, we start with an original DNA molecule. You can see that up here on the top. And that molecule, first off, has to be what we call denatured. This is going to be done by heating it up to really high temperatures so that we can separate the strands, which requires breaking of the hydrogen bonds. So let's not forget that that's what's holding the 2 strands together. So we break the hydrogen bonds using high temperatures. And then once we do that, we're going to have some other DNA molecules inside of our test tube that match up with specific sequences on our DNA. And these little molecules are called primers. So you can see that right here. And so they're shown in yellow. And so we're going to lower the temperature now a little bit so that these are able to actually stick onto the DNA sequence. And so we call that annealing. So the annealing of a short piece of DNA to the ends of the sequence that we're interested in is going to be the next step. Once those have annealed, then we've got a starting point, and you can see that starting point down here. So these little fragments, they're providing the primer, so the starting place for our DNA polymerase. And DNA polymerase is then going to jump on there and it's going to extend that end so that we get just the piece amplified or synthesized that we're interested in. Other areas on the DNA, there's not going to be a primer there, so DNA polymerase is not going to actually make a copy of that specific region. So there's 3 main steps involved in PCR. We have denaturation, separate the strands, then we have sticking on of the primer, and then we have the actual extension, which is the making of the new DNA. In the end, this is what we end up with. First, we start with one molecule. After the first cycle, we have doubled that. Then if we run another cycle, we've doubled it yet again. And then you can see that with each cycle, we completely double what we actually have inside of our tube. So yeah, we can end up with billions of copies if we're doubling every single cycle. And typically we're running maybe 30 cycles, maybe 40 cycles. So numbers get really huge as far as copies that we have inside of that cell at the end. When we run all these different techniques, one thing that we're going to have to do is we'll have to be able to select a clone of recombinant cells. And so to do that, we have a number of different ways. One of them is going to be with the use of probes. Probes are DNA sequences or RNA sequences that selectively bind to complementary sequences. So they're going to have that complementary sequence, they're going to stick to whatever matches. And these probes, we label them with either radioactive markers, sometimes it's a fluorescent marker, but then they will stick and we can specifically see exactly where our gene or our sequence of interest is at. Another way that we select is with the use of gel electrophoresis. This is going to be separation of molecules based on charge, size, and shape. And the apparatus that we use for that, you can see part of it here in this picture. So we will have a gel-like matrix, so it's kind of like Jell-O, and we will put our DNA sample in there, and then we will look at how that DNA is able to move through that matrix in response to the charge that we apply. And typically, it's like an obstacle course. A tiny molecule is going to be able to maneuver through the matrix much faster than a large molecule. It's going to be much slower. So we're getting basically a size separation here. So we can see on this end, what we are calling the wells, this is where we place our sample. And so this would be the sample that contains a whole mixture of DNA. And then we apply the charge. DNA molecules are negatively charged, so these DNA molecules are going to move towards the positive end. And so down here is where we're actually applying the positive charge. So our DNA molecules are going to be attracted to that, and they're going to start to move through the gel. Again, the tiny ones, they're going to move a lot faster. So the marks that you see here, the different bands that we see, those are representing the DNA molecules, and they would have to be labeled or stained in some way that we could actually visualize them. Now, if we combine a few techniques, we can get what we call a southern blot. A southern blot is going to be taking DNA from gel electrophoresis, so separating the molecules similar to what we just looked at, and then moving them onto some type of substrate that's going to be easier to manipulate them. It's going to be a membrane, a kind of a piece of paper, and then we're going to take that piece of paper and we're going to incubate it with a probe. So we're really combining the gel electrophoresis and the probe together for southern blots. If we look at the general idea, we've got the first column here, which is pretty much showing us the taking of the DNA and running gel electrophoresis. So we're going to take a big DNA sample, we're going to be incubating it with a restriction enzyme to cut it into fragments, and then once we get the fragments, we're going to use gel electrophoresis to separate them. So we've got this collection of fragments of all different sizes inside of our gel, and the gel is very flimsy, it's hard to manipulate, it breaks easily. So we're going to take that and we're going to transfer it onto some type of membrane, which is underneath here. So the membrane is going to be similar. It's nylon, so it's kind of like paper, a little bit like fabric. It's something that's not, if we can pick it up, we can move it around, we can incubate it with liquids, and it's still going to hold its shape. So we're, in this case, what we're doing is we're transferring the fragments onto that membrane. And so the membrane is right here. The fragments start in the gel, and then over time, we're basically moving them down so that they end up stuck onto the membrane. And so when we look at this picture up here, this is the membrane with our DNA fragments on it. We're then going to take the membrane, we're going to incubate it with a probe. That probe is going to stick only to the sequences that we're interested in, and then we look at where the probe is at. That's going to depend on what we've labeled the probe with. Sometimes it's labeled with radioactivity, so we can look at that using some X-ray films. There's also some non-radioactive means or fluorescent means to actually identify where the probe is at. So that's the southern blot. You can see that when we look at the picture at the end, we can see that out of all of these DNA fragments, what we're interested in is really only showing up in a couple locations. Other techniques for selecting specific clones, DNA microarrays are going to be a way to look at a ton of different molecules at the same time. This is going to contain many immobilized DNA molecules, and these are spotted by some type of robotic machinery, typically onto a glass slide or a chip. And when I say many, we're talking thousands, hundreds of thousands sometimes, onto a tiny little chip. And this is kind of the overall idea of what it does. So we take lots of DNA sequences, they are digested, they are then spotted onto this glass chip, and so they're fixed in a particular location. Then we take our probes and we basically incubate the two together, and then we detect them. In this case, you can see that the probes were fluorescently labeled, and so we get different colors based on how much actually stuck to the different spots that we did have on our microarray. This is used to monitor gene expression, diagnose infections. It can be used to identify specific organisms. a lot of usefulness to this, and the main reason why we're able to do this is because we do have a lot of really good robots, robotic techniques that can be used these days to not only just spot things on there, but also to detect what we end up with. Because we're looking now at thousands of genes at a single time, and it does require the use of robotics. It does require the use of some computing power as well. When we get down to actually sticking our DNA sequence into cells, there's numerous techniques that we use for that. One of them is going to be electroporation. So electroporation is basically going to take our vector and we're going to get it into the bacterial cell. This is going to actually use an electrical current to actually poke little holes in the outside of the bacterial cell, if that's what we're actually using. So notice that we've got tiny little holes around the outside. Then we actually put our DNA there with it, and we hope that the DNA enters the cell, and then the cell basically patches up the outside. They can certainly recover from the making of the holes, so we're just temporarily damaging the cell wall and membrane so that we can get our DNA fragment in there. And then by the end, if you look at that last picture, you've got your DNA piece actually inserted into this cell. We can also have what we call protoplast fusion. With protoplast fusion, basically the cell wall is dissolved, digested, we get rid of it so that we have what we call a protoplast. So the protoplast is the cell without its protective cell wall. Then we take the two remaining protoplasts and we fuse them together. So we have to use chemicals to get them to actually fuse and connect together. So what you've done is you've taken two cells and you've combined them into one single larger cell, and that ultimately is going to fuse the two genomes, so fuse the genetic information in the two cells together, and then the cell rebuilds the outside, it rebuilds that cell wall. So that's just another way of combining the DNA together. And another one is what we call injection. So this is actually firing, sticking, physically putting the DNA into a target cell. Now, a lot of times we take some type of bead, metal bead, and we coat it with the DNA of interest. And you can see in the first picture, we have what we call a gene gun. It actually shoots these particles that have the DNA on them at a cell, and hopefully they force those DNA particles, metal beads coated with DNA, they force them into the cell, and then hopefully that target cell takes the DNA and incorporates it into its genome. So that is a physical forcing. The other slide picture that we see here is microinjection. You would have to have a very tiny pipette that's able to take the DNA and puncture and insert that into the nucleus of your target cell. If we look at some applications of recombinant DNA technology, genetic mapping is one of those. With genetic mapping, we are locating genes on a nucleic acid molecule. So we're figuring out exactly where our gene of interest is located. One way that this is done is using fluorescence in situ hybridization. In vitro was a term that showed up earlier. In vitro is artificially in a test tube. In situ is real life. So this is actually looking at where the gene is at inside of an organism, inside of a cell. So what this one does is it takes a probe, again, that has been specifically labeled, so a sequence that's going to stick to complementary matches inside of a cell, and it's been labeled, so we denature our sample, we incubate it with the probe, and then we look at where it's actually sticking. And as you can see in this picture, we can see on this chromosome exactly where our gene is located at because it's showing up specifically in red. Now, if we're doing this inside whole cells, you can get a really nice idea of where that gene is actually being expressed, where the organism is actually using the protein products at. So that's what we're seeing here. This is like a little embryo, and we can see that we've got specific bands where this protein is being expressed, being made. That's one look at it. We can also see it right here. We can use a couple of different probes that produce different colors, and we can see different regions inside of an organism where this protein is being produced. Another way that we can use genetic mapping is with automated gene sequencing. So this would be to determine the gene sequence of an organism. And so that might be the gene sequence of a plasmid, that might be the gene sequence of a viral genome. And so this does typically involve some type of electrophoresis, so separation of fragments based on size. It's also going to involve some use of fluorescent nucleotides so that we can see what the order of the different bases are within our DNA sequence. There are many pharmaceutical and therapeutic applications as well. One way that we can use recombinant DNA technology is with protein synthesis, so protein production. We can make large quantities of proteins. We can make a variety of vaccines. We can do some genetic screening. If we're doing genetic screening, we can look at basically what's different about one individual versus another individual. Do they have what we call a marker that would predispose them to having a specific genetic disorder? DNA fingerprinting, basically looking at an individual's DNA sequence and how it is different from everyone else's DNA sequence. So you might have something similar to what you see in this image here with that. We can do gene therapy. That is a way to treat specific disorders. And then we can also do a variety of medical diagnoses using recombinant DNA technology. Another one that we have is xenotransplants. So that is basically using organs from other types of organisms and putting them into human beings. In this example here, you have a kidney that has been grown in a pig. So we use a pig donor, and then ultimately we place that into a human being. And there's a bit of kind of cloning that goes along with all of this to help get both the kidney and the pig ready, and then also the human being ready, because we do want we want them to be able to accept each other. We don't want to have some kind of immune response to that particular organ that we put in there. So using the recombinant DNA technology is going to help us to minimize the chance that that particular organ that we're placing into the human, that it's actually going to be attacked by the human's immune system. We can also have a lot of agricultural applications. Just a few of the examples: we can produce herbicide tolerance in plants, so we can make them tolerant to weed killers. So in this way, we could take our crop of interest, make it tolerant, and then if we just spray the herbicide on that crop, we're selectively killing the weeds, even though it happens to be getting on our crop as well. We can engineer them for salt tolerance, so they can be grown in greater regions. We can engineer freeze tolerance. Also pest resistance, so help them to resist various insects, various maybe viruses. And then we can get overall improvements in nutritional value and then also yield. So this would make our crop even better for those who are actually buying it and eating it.